Our visual experience begins when the visual pigment in our retinal photoreceptors absorbs photons of light energy, ultimately leading to the closure of cyclic nucleotide-gated channels in the cell membrane. This suppresses the release of the neurotransmitter glutamate from both rods and cones. The released glutamate is sensed by downstream retinal cells. To study the downstream signals, the eyes are removed from a euthanized mouse. The retinas are isolated, embedded in an agar block, and sliced using a vibrating microtome. Next, the retinal slices are placed under a microscope and stimulated with LED lighting while patch clamp recordings are taken to measure the synaptic currents or potentials generated.
Generally, individuals new to this method will struggle because of the added complexities of working in the dark, so being able to visualize the entire process will provide a feel that's otherwise difficult to describe.
Begin this procedure by coating the recording and reference wire electrodes that will be used with a layer of silver chloride. Dip the electrodes in 1 molar sodium chloride, then using a 15-volt power source with a 1 Hz sine wave, drive 15 volts between them for about 20 seconds. Using a micropipette puller, prepare patch pipettes from fire-polished borosilicate glass, choosing a pipette tip resistance based on the size of the target cells. For cell bodies about 20 microns in diameter, use a pipette with a tip resistance of 5 to 10 megaohms. For cell bodies that are about 5 microns in diameter, use a tip resistance of 12 to 15 megaohms. To prepare the ground reference electrodes, use a syringe to fill polyethylene.